So Eric asked me to talk about preprocessing, so that's what I'm going to do. I'll get right to it. Okay, so the main thing I want to do is explain what preprocessing is and why it's important and why you want to understand it, even if you're not going to be developing preprocessing methodology. But to do that, I have to explain a little bit about the technology. You've seen a little bit about already, but I'm going to go a little bit into some more detail. And to make the point of how this could be important, I'm going to show some examples. I'm going to stick to expression, gene expression, but there's examples in other platforms, other applications as well. I do have slides, but I'm not going to get to them. Okay. And by the way, this is different from what you have on, a little bit different from what you have on your booklet. Sorry about that. Okay, so you saw this already, the central dogma of biology. And basically what I wanted to point out is that the technology, microarray technology, and sequencing technologies as well, is what it does is it provides high throughput measurement of various of these outcomes like RNA or gene expression, but there's protein and DNA and all that kind of stuff. But I'm going to focus on gene expression array. But just to give you the big picture, what we want to do is measure gene expression for all genes, or all known genes, or all genes that the company decides to put on the chip. Okay, so to motivate my talk, I'm going to use 3 examples that I'm going to use to motivate the importance of understanding data analysis. So I know that when I give this talk, or when I write my papers, many times I think people don't see how important it is, and people use their own methods, or they buy commercial products to analyze data, or they don't have anybody in their lab that understands data analysis very well, and they publish, right? So they think they don't need it because they get to publish. A lot of these papers are never found. You never found mistakes with them, but they don't get cited very much either. So the results may be wrong, they kind of disappear. But some are lucky enough that some statistician, like Keith, for example, finds a problem with it. And then those make headlines. So you don't want to be one of these people. You don't want to be one of the papers where you describe a result that is wrong because you didn't do data analysis right. That's what I want to keep you from doing. And the best way to do that is to understand, at least conceptually, what happens in the data analysis. And that microarray data analysis and sequencing is the same. It's even more complicated, but it has a lot of steps. I'm focusing on just the first one, which is what I call preprocessing, and I'll say more about what it specifically is. The 3 examples I'm going to talk about are the first one. This is not a paper. This was just conventional wisdom back the beginning of the 21st century, that Affymetrix arrays were too noisy, and a lot of people were making their own because they didn't want to use these arrays that were, when I say noisy, I mean there's a lot of variability when there shouldn't be. I'm going to show you that is actually not because of the technology, but because of the way the data was analyzed. The second one is this paper that claims that in terms of gene expression, any human tissue is more similar to another human tissue than when you compare it to a mouse tissue. So think about what they're saying. They're saying if you compare a human eye to a human testicle, they're more alike than a human eye and a mouse eye in terms of gene expression. I'm not a biologist, but to me that doesn't make any sense. I'm going to show you how they made that mistake. Again, it was incorrect data analysis. You take the same data, analyze it right, you don't get that answer. The third one I'm going to talk about is this paper that claims that 70% of genes are differentially expressed between whites and Asians, specifically the HapMap CEU group and the ASN ethnic groups. And that also, I'm not a biologist, but that also doesn't make a lot of sense to me. And as you'll see, it's also a consequence of bad data analysis. And then tomorrow you're going to see what bad data analysis can really do harm when Keith Baggerly speaks. Okay, so pre-processing is the first step. I'm not talking about much of what comes after, which also can introduce problems. I'm talking about just the first step, pre-processing. So what does that mean? I'm going to go little by little and get you to what that means. So this is what gene expression data analysis used to be like. You can see here that you consult with a sophisticated statistician and they tell you that this tissue is expressed in this tissue and not these tissues. This is a northern blot, I think this is, and that's what it was. So I'm sure this just to point out that biologists didn't rely on data analysis much before, but now they need to. This is gene expression, just like the previous picture, but instead of one gene, this is just 2 samples, and it's 12,000 genes. That's what each point is here. So these are 12,000 genes. Sample 1, sample 2. In the previous picture, there was about 8 different samples. This is 2, just 2. So how do I analyze this? Which genes are differentially expressed between these 2 samples? So I'm going to focus my talk on differential expression analysis because it's maybe the most widely type of analysis that's done. There's other things you can do. You can do class discovery, class prediction, but I'm focusing on finding genes that are differentially expressed in, to keep it even simpler, 2 populations, like Asians and whites, for example. All right, so we have a bunch of genes, and now how do you do the analysis? Which genes are differentially expressed here? How in these 2 samples? So what do you do? What do we do if we want to find out? What do you look for? Think about it for a little bit. If you're not a mathematician, what is a gene that's different in one sample versus the other? You have to be far away from what? From the diagonal, right? So this red line is a band around the diagonal. So genes that are outside these bands are twice as big on one sample compared to the other. That's what they are. Okay. This is in the log scale. All my data is shown in the log scale, log expression. There's reasons for doing that. One of them is that biologists are interested in fold changes. How many times more bigger is the expression on this sample compared to this other sample? So if we want to do that, we look at that. But there's actually something better we can do, which is to rotate the data 45 degrees. This is something Terry showed me 10 years ago. And when you do that, you have the log ratio on the y-axis and the average intensity on the x-axis. So we have 2 measures that we are interested in on the axes of, the y and the x-axis. So same data, just a different look. And now, which are the genes that are different? Those that have a big log ratio, right? So the genes that are out here and the genes that are out there, right? That's what we look for. So now it's easier than before because instead of turning our necks and looking at it like that, we can turn the picture instead. Okay, so that's what that is. So now this is one of my first Affymetrix data sets. These aren't 2 different tissues. This is the same RNA on 2 chips. So these are all these genes that we see out here being called differentially expressed, if you just go by size of fold change, are all mistakes. If you pick genes that are twice as big on one sample versus the other, you get about 2,000 out of the 12. And they're all mistakes, without exception. So this picture was what made people stay away from Affy for a while. And the biologists that I was working with, when I said, what's this? We can't use this data. And their response was that biology was like that. When there's low expression, notice that the wide range of the variability is bigger in the low expressed genes with low intensities. This is the average of the 2. And you can see there's more out here. But once we look at the manual, we see that maybe it's not the biology, maybe it's the statistics that are done, and that's the pre-processing part. So there's a lot of steps between the first raw data and when we get to here, and I'm going to explain a little bit of that now. Okay, any questions? Okay. So we need to understand the technology a little bit to understand why this is happening. So, very quick introduction to molecular biology, super fast. Nucleic acid, there's 2 strands, and you can separate it, you can denature it, and when hybridization occurs, what that means is if you have a single-strand DNA and there are various other single-strand DNAs around in the vicinity, and they're in some fluid where they can move around, only the one that is complementary, which, can you tell which one it is? You know enough biology to see it? ATG, this one, right? So that one is going to stick to that one. That's the big trick. So if they're floating around, that's what happens. That's the trick that makes microarrays work. You'll see in a second. So for gene expression, you have DNA. Thanks to the efforts of the 90s and a little earlier, we're sequencing the genome and doing other things. We think we know the sequence of many genes. So we know that this is a gene, and this is the sequence. So when an RNA transcript is transmitted, we know what sequence that is going to be. So for all 20,000 genes, or 30,000 or whatever we're saying there is, we know the sequence that the RNA is going to have. Then there's another nice trick biologists came up with called reverse transcription, where you can turn RNA into DNA, and you can actually make a lot of copies of it if you want, this is nice because our DNA is more stable than RNA. What this lets you do is have you have the RNA turns into a DNA, and now you have these pieces of DNA that are single-stranded, and if you construct some piece of equipment where you have the complement, all these guys will stick to it. So this is the basic idea of microarray technology. We have these RNAs floating around, or we want to know if they're floating around. Remember, this is something we don't see in a microscope. It's in some cup, and there's some liquid in it, and the biologists tell you that there's RNA there, but you can't see it. So we want to know which genes it is, the RNA from which genes are floating around in there. And by the time the biologist is going to use an array, it's actually turned into DNA with this previous trick. So they're in there. Which ones are there in there? That's what we want to know. So we can't see them, but we know their sequence, right? They're in some database, and I just made it up here. So that I went into a database and I said the gene I care about has that sequence. If it were turned into DNA. And what I'm going to do is I'm going to construct on a piece of glass. I'm going to synthesize the complement of that, right? So down here, see, I constructed another molecule that is the complement. So if that gene is actually there, and it sees these guys, they're gonna stick together, and that's the lobster trap that catches the lobster unless you count them. So that's what microarrays do. They take, instead of just that one gene, they do it for all genes. You have this piece of solid that's about a centimeter squared roughly, and it's divided. These days they're divided into 6 million little squares, and in each one of those little squares or cells, you synthesize millions of copies of a known molecule, and you keep track of which molecule is in which square. So by doing that, this is a simplification where you just have 4 instead of 6 million. You know what sequence this is, you know what sequence that is, you know what sequence that is, you know what sequence that is. Here's array 1, array 2. These are the gene transcripts that are floating around in some sample. You want to be able to count them. So you label them with little suns, and then you let them hybridize to the array. So now you have these fluorescence things emitting photons that are stuck in a certain place. So with the scanner, you can turn this into this, and you know what sequence goes with what square, and you can count it. You can count how much light you saw. So now I know there's 4 of the blue, 2 of the red, 0 of the light blue, 3 of the green. On the other sample, it's different, and if I'm comparing, say, normals and tumors, I would say, well, look at this one. There's more in cancer than in normal. Let me write a paper. Okay? So that's how my arrays work. Of course, it's not 4, it's millions, and this is what this picture actually looks like this, where each one of those little squares is a different molecule, representing a different molecule. So that's the original data. It's a big image with intensities in each little square. So you have 6 million numbers, and they somehow get turned into 20,000 gene expression estimates. How does that happen? That's what I'm going to talk about. So let's get back to this question. Why is there so much noise in this array? So now I'm adding something to the picture, but let me review what it was. This is the log ratio. On the y-axis, I'm using m, and it's in log 2. So this is, the red means 1 or more, minus 1 or less. So that means twice as big. It's log 2, right? So 1 means 2. So it's twice as big on one array compared to the other. Either array 1 versus 2 or array 2 versus 1. Right? So so I have 20,000, what was I saying? There's about 20,000 genes here, represented here, so 20,000 points. And you see all these red ones I've marked because they have this fold change is bigger than 1, and they're wrong. So there's about 2,000 of them. But now I've added something else. I've added these green numbers. Those are also genes. There's 16 of them. And these are genes that we were lucky enough to get this experiment early on, where a company spiked in, that's the name that they use, they put in known amounts of these genes into the array. So for these 16 genes, we know they should be different because we made them different. Twice as big. The green ones should be twice as big on one array versus the other. But all the other points should be the same because this is the same RNA. It's the same RNA except for these 16 genes. So we should see these separating out from the signal, but we don't. We see all this noise, and these guys are you can't find them. So if this is what Affymetrix is producing, then people are right. We're right to be saying this technology is too noisy to use. Okay, so preprocessing. So what I'm claiming is that noise is due to statistical manipulations and not to biology and not to technology. So I'm going to talk. These are the steps of preprocessing. This is what we do when we go from the intensities to the actual numbers that are associated with expression values. Normalization, background correction, and the last step, summarization. Notice I've been saying there's 6 million, or there's from hundreds of thousands to 6 million intensities you start with, and then at the end there's only 20,000 genes. That's because there's redundancy, and that's what summarization is. But today I'm not talking about that. So I'm only talking about the first two. So normalization, what's that? All right, so here are box plots from 6 arrays. These happen to be replicate arrays. Did I say 6? Well, that's because there's 3 kinds of statisticians, the ones that can count and the ones that can't. So these are 8 replicate arrays, and they're color-coded because they were done in different dates. But they're all supposed to be the same, and you can see that there's big differences. You can kind of see the difference between dates, the green and the orange. But even within the same batch state, that you can see that there's this big difference. This one appears to have 4 times the expression as the first one. That's a very common phenomenon that you see in these data. You take RNA, you split it into 2, you put it on 2 arrays. And you think they should give you the same thing, but you end up seeing this. Very common. So this is one of the reasons why arrays are, I don't think ever used. Maybe someone has tried, but they're rarely used to say this cell or this group of cells had more expression in general than this other one. Why? Because when you think you're putting in the same thing, you see that as well. So this is fortunately pretty easy to fix statistically. One of the things that we've done, a lot of different people have come up with tricks that are similar, but the main idea is to assume that different samples should have globally the same properties. So maybe some genes are different, but globally they should be the same. That's a key assumption. We've made it for 10 years now, and it seems to work. So you can statistically, this is not saying the biologist, I can't use this data, do it again. I don't care if it was where it would cost 1, 2, 3, 4, 8 times 500. You don't do that. You try to fix it statistically, and it's very easy to do by forcing these distributions to be the same. Right? So you just make them the same. Right? That's what we do. And then you proceed. Right? So that's easy to do. For example, you could subtract the mean and divide by the standard deviation. That's one way you could do that. But that's not a very good one, but it's better than not doing anything. And there's many papers written on different ways of accomplishing this. Okay, now, is that the problem? Is this why this data is so dirty? It's not, because this data has already been put through a process like that. So that was seen right away by everybody. Everybody did something to fix it. So this has already been normalized in a relatively okay way. So then what is it? So let's look at the next part of preprocessing and see if that's the problem. It's background correction. Okay, so why is this background correction? Why do we need it? So this is real data. These are box plot again of all the intensities of gene expression for 6 arrays. And this is an experiment that I did. I have biologists I'm friends with that spend about $1,500 on experiments like this, where we put water on a chip. That's what this one is. We put DNA with no label. We put yeast DNA on a human array. We put poly G and poly C. All right. So this is an experiment where the RNA that you're putting on the chip is not represented by the array. So no intensity should show up. Especially when you put water. You put water on a chip, you should not see anything, but you see a distribution. It starts at 32. Most everything's at 32, but then there's some big ones. But more interestingly, when you put yeast DNA that doesn't match, so there is actually DNA, but it doesn't match the chip, you then see much more in terms of intensity. So that's what background correction is needed, because even when things should not stick to the probes, they do. Probes, I think I forgot to mention that, it was in the slide, but probe is what we call the group of molecules that are targeting, that are hybridizing to the RNA. So we have thousands and thousands of different probes targeting genes. Okay, so why do we background adjust? So this plot is real data again. And by the way, I should stop saying that. Everything I show you here is real data. I don't have one simulation anywhere. It's all real. So I won't say real data anymore. Okay, so this is from this experiment where we spiked in known amounts. And what I'm showing here are the average of all the probe intensities that were not spiked in at all. So there were some that were left out, that were spiked in at one half of a picomolar, 1/4 of 1 micromolar, 2 picomolar, etc. So one good thing about this picture is that as you augment the amount you purposely put in, the intensities also go up. If this line was flat, then what would have happened to Affymetrix? What would have happened? They would have gone out of business. So we do see a line. But one of the problems we see with that line is that it starts at, in this case, 180 or something. It should start at 0 because there's nothing to be found. But why doesn't it? Because there's background. So how do we fix this? Why is this a problem in the bottom line, in the actual answers you give? Well, let's go slowly on this one. So if you are going to report a fold change, then when you're at the low end of the scale, then you are going to be reporting the real expression plus some background divided by the real expression plus some background. And if these backgrounds are big, then this ratio is going to be about 1. Even if these two are very different, like in this case, this is twice as big as that. This is in the log scale. So this guy's twice as big as that, but we don't see it go up 1 because there is background. At the higher level, it's not as much of a problem because these guys, these E's, these expressions are bigger than the B and they overwhelm it. So up here it's not a problem, but down here it is. So if you don't background correct, then all the intensities that are in the low range, if there is a real difference, you're not going to see it. So, that's something that the company apparently did not want to happen, and they went through a lot of effort to fix it. So how do they actually spent half of their chip, they dedicated to fixing this problem. So when you paid $500, you were 250 of those were to fix that problem. So what they don't do this anymore, but they used to, not so long ago. So how do they fix this problem? So let me explain a little bit more about how this technology works. This line up here is representing an RNA transcript. It's 3' and 5', and it's about 2,000 or so base pairs long. And I want to design probes and put it on the chip that hybridize to that transcript, right? So I know what transcript is, so I know what sequence to put on. So what I do is I pick, some complementary molecule to this piece of the transcript, and I put it in some cell on the chip. Affymetrics, because of the way they construct arrays, they can only make molecules that are 25 base pairs long, right? 25 bases long. So in order to have a better representation of the gene, they actually put down 11 different molecules to represent one gene. So this is the first one, second one, right? And then that's represented down here. So this particular one I'm using as an example, here's the transcript. So I'm going to take the complement sequence and I'm going to put it here. And because this gene is present in my sample, this thing gets very yellow. And then for this other one, I'll have another transcript, so I have a bunch of intensities. Now, one of the things I noticed was that, and I'm going to get to that, is that every one of these molecules had a different way of having background effects, and I'll show that in a second. So they constructed a special probe just to measure background by changing the middle base to a G. So that would be put on another place on the array. And then what they would say is, well, this one's measuring background plus what I want to measure, plus the signal. The other probe is measuring just the background. I'm going to subtract it, and I'm done. So why were they spending one probe for each probe? Why were they adding another one? Right? Why not just estimate some general background and be done with it? So here's the reason. And this is going to come back again later. The reason is the probe effect. What is the probe effect? Okay, so we're measuring RNA, and we're using different probes to measure the same RNA, and then we're using another set of different probes to measure another RNA, etc. Each one of these probes is a different molecule with a different sequence. But one of the consequences of that is that they result in different intensities, just because of their composition, it turns out, apparently. So here is an example. Here's the data showing that. These points you see here are the intensities, the observed intensities for, in this case, 16, or is that 11? 11 probes associated with a specific gene. So this is one gene, one array. And these are the 11 intensities associated with that gene on that array. Now, it's the same gene they're measuring. The answer should be the same for all of them. But you see that, for example, this one is about 50 times smaller than this one over here. They're measuring the same thing, but they're giving you completely different answers. However, that sounds like it's terrible and that's just going to ruin everything, but it turns out it's not as bad because when we measure again in another sample, we see the same effects. And what I'm showing in blue is the same gene, but on another sample where it was twice as big. I know it because I put it in. And what we see is the same pattern again. So it's a very consistent pattern. So even though probe by probe, the information is very different. Across arrays, it's very similar. So we are able to notice that this gene is twice as big in the blue sample compared on the red sample at every single probe. It's very, very good at that. See, every time it's about twice as big. So you don't look at the absolutes, you look at the differences. Okay, so that's the probe effect. Now, how does it affect background? Here in this picture, I'm now showing the same one. This is one gene. These are the 11 probes for that gene in different colors. And now I'm showing the picture where I have different arrays at different, where that gene was spiked in at different quantities. And here's the observed intensity. And what you can see is that at -2, which is -2 log in the log scale, is a picomolar is basically not there. It's not present. It's background. Look at how much the background changes between probes. So here's the problem: is there's background noise, and it's different for every probe. So you have to fix it for every probe separately. That's why they said, let's just spend half the chip on this. So their approach to fixing this problem was to estimate these numbers. This is 11 numbers times 20,000 genes. It's 200,000 different backgrounds they have to estimate. So they had a specific probe for each one. So this is their approach. We want to estimate the expression for one sample, but we're always interested in the difference between two samples. So we really want the difference between sample 1 and sample 2, the expression. But what we're seeing is background plus expression divided by background plus expression. So their approach was to estimate B with a probe, and then they would look at this. They subtract this B from that B and that. This is the observed B, and this is the true B that they want to get rid of. Now, I'm not showing you the math behind this, but I could if you wanted me to. But this transformation you can show it mathematically using Taylor's expansion, for example. The variance of this creature goes to infinity as this thing goes to 0. So that's what the picture showed. So that's not biology. That's math. So one of the things that I did in conjunction with Terry Speed and others was develop a mathematical solution that didn't have that property. Instead of using this, you design a mathematical solution to the problem of estimating this, given all this information, that didn't have that property. And what ends up happening in practice is that we turn this picture, without changing the date, doing the experiment again, just changing the math, we turn this picture into this picture. And at first, people didn't believe it. They thought I had taken out genes and all that, but it's really actually pretty simple to see why it happens. So that's the first story. So the Affymetrix technology is not that noisy. And once this was out, and other procedures like this, I think Affymetrix started selling more chips. Okay, so that's lesson 1. You do the data analysis right, you can turn a noisy technology into a very decent one. Okay, second one. Now I'm going to the paper that claimed mouse and humans were... Mouse liver is more like human brain than mouse liver. All right, so this plot is showing you. So imagine we now know how to fix Affy. We can take an Affymetrix arrays and get a nice estimate of expression for each gene for each array. So for this picture, I downloaded 500 arrays from the public domain from different tissues, different studies, and I computed this vector for each one. So I have a 20,000 by 500 matrix where each column is a different tissue and each row is a different array, and I compute the pairwise correlations between each tissue. Right? Okay, so when I do that, I get that everything, pretty much the correlation between anything is above 0.8, 0.9, near 0.9, sometimes as much as 0.99, no matter what it was. So in this picture, I'm showing you pairwise correlations between two arrays, or which represent two tissues, and sometimes the same tissue. But you can see, so any point here, if you pick a point here, blue means
dark blue means 1, white is 0, and red is negative. You don't see any red, you don't see any white. It's all high. So why is that happening? Why is the correlation always high? And actually, this is something that AFI and others use to sell their products. They show you the correlation between replicates. It's always like 0.99. But if you take a liver and a kidney, again, the correlation is also going to be 0.90 something. So don't be fooled by that marketing ploy. So why is that happening? Why is everything so correlated, no matter what? Well, it has to do with the probe effect. So let me just show it in a closer scale. This is, I'm showing you a gene, 2 genes, gene NM00315 and gene AW276646. These are 2 genes. I have one sample, and in that one sample, I saw that gene expression estimate given by the array was 8 in both. So both genes say 8. What does 8 mean? I don't know, but they're both 8. So if I ask you which of those 2 is more expressed in this sample, this gene or that gene, if you don't know anybody, you'll say, well, they're both 8, so they're roughly the same. Right? They're roughly just as expressed. Okay, now I'm going to add something to this picture. I'm going to show you the histogram of all the expression for that gene across all these tissue arrays that I got. And look at what happens now. So in this case, in this gene, almost all the gene expression is down here at 4. And then there's some that are above 5, very few tissues above 5. In this case, almost all is around 8 or 7, and there's a few above 9. When I see this picture, I tend to think this is all these tissues here, the gene expression is 4, that's basically equivalent to not being expressed. They're not expressed, and that gene is expressed. That gene in that tissue is expressed. In this case, the background ones are up here. They're not here. And we already saw a hint of that. When I showed you earlier, different backgrounds are different, so even if we try to fix it, we really can't. It's very hard to fix it. It survives my background correction, and we see this big discordance. So, what does that say? That tells me that this gene, even when it's off, it's high. And this gene, when it's off, it's lowish. So if you take the correlation if this is true for all genes, and it is, and you take the correlation, it doesn't matter what's happening biologically. Some genes show low intensities, others show high intensity. It has nothing to do with what's actually happening. So of course they're going to correlate, no matter what. So that's what the probe effect does. And why is it happening? Because a different set of molecules is used to estimate this expression and that expression. And that chemistry has a big enough effect to make this happen. So that's not that hard to fix, though, right? Statistician, we can fix it. If we have this data, we would center it or something. Right? So that actually does help. If I do, this is before centering, and then I do the same exercise of computing pairwise correlations after centering, I get this. Now there's a lot of white. That means kidney and brain are not correlated, as we were saying before. And then there's some pockets of blue. That's because this is, for example, that's a study, and they're all some kind of blood. And this square is all from the same study, and there's some kidney. These are all some liver, these are all some brain, etc. So now, tissues that are the same are correlating, tissues that are different are not. Okay, now, back to the mouse and human being more alike. So now I'm going to take human tissues data and mouse tissue data, and this is real again. I said I wasn't going to say it, but I said it again. This is from the internet. We got mouse and human tissue data, and we computed expression vectors for each one, and computed the correlation, and you get this. So that's what the paper was saying. These are humans are blue, mice are red. It doesn't matter what tissue you are. This is a dendrogram showing the results of applying a clustering algorithm to the data. So lung and spleen from humans are very alike and very different from lung and spleen from mice. Now, why is that happening? It's a little subtle, but if you follow closely what we've been saying, you'll see that the reason is that it has to do with the probe effect. The human chip for gene 1 uses one set of molecules. On the mouse chip, it's a different set of molecules. So the probe effect will be different on human and mouse. So for human, it really doesn't matter what's happening. Gene number 1 will always be fluctuating around 6 or 7. On mice, it will be fluctuating between 6 and 7. So in mouse, they're always the same. In humans, they're always the same, regardless of what's actually the movement that actually matters. Now, I should ignore the barcode thing up there. If I center the data, like I just described, just that simple exercise, if these people that wrote this paper had just known about the probe effect or had read about it because it was written quite a bit on it, they would have subtracted the mean or something and divided, but standardized in some way, and they would have gotten this picture. Brain, brain, skeleton, skeleton, liver, liver, kidney, kidney, lung, lung, spleen, spleen. Just that. That's all they would have to do to not make that mistake. All right, last example. Asians, super different from whites. I'm going to use this to explain why that happens as well. So this is the after centering plot. And this makes sense that you have these blue squares in the diagonal because those are the same tissue. Studies, unfortunately, for us statisticians wanting to do meta-analysis, studies are correlated with tissue. That's not the biologist's fault. If you're interested in liver, you're going to study liver, and that's what your study is going to be on. If you're interested in kidney, you're studying kidney. So a study is usually all the same tissue. But there's one exception down here. This one. Is it my next slide? Yeah. Yeah, this is exception. This is a study that a pharmaceutical put together where they just did a bunch of tissues in one study. So it's one study, different tissues. But we see that they're all blue. So even though there's liver and brain and testes and all kinds of tissues, this is all correlated more than, say, with other studies. Why is that happening? Okay, and here's a close-up. Right, so you can see let me see if I can read some of these. Ovaries and spinal cord are blue, so they're correlated. I know most of them are. All right, so now this is the picture I showed you earlier. And now what I'm doing here is I'm taking the same data and I'm stratifying it by tissue. So you see there's two tissues where that gene appears to be on, and the rest, it looks like they're off. And then in this gene, two tissues looks like they're on, and the rest, maybe a biologist wants to argue with me, but I would say they're off. So these two are on, these are off. Now, if you look closely, if you do a t-test between these numbers and that number, what's the p-value going to be? I think you said it earlier. It's 10 to the minus 18. Was that right, Robert? 10 to the minus 22 or something like that? Well, in this case, it is going to be that because these are all bigger than those. Okay, so but I'm saying they're not expressed. So they're not expressed, but they're different. That doesn't make sense, right? So I think that that's something to do with batch effect. That's the study, the lab, whatever, is making these things vary and not the biology. Now, one thing you can do to test that is that I can turn all these into ones, into zeros, and leave these as ones. Just to test that, right? Because now these won't be different anymore. But then I still conserve some of the biology. And if I do that, this picture turns into this picture. So now all this blue that was here is gone. And there's one little square left down here. So did it not work? Well, look at what these things are. This is spinal cord, cortex, pons, lobe, bulb, loba, all brain. So this is what we want. We want the brains to be like each other. All right. So, batch effect. It's a big problem, and not everybody knows about it. And if you don't know about it, you might publish a paper with the wrong result. Here's data that was original, normalized. They look like it's fixed. However, about 200 to 1,000 of the 20,000 genes follow this pattern. Even after I've corrected them like this, a few still are different. So even applying the strongest normalization leaves some genes that they just don't get fixed. And it probably has something to do with background levels being slightly different, maybe. I mean, see how these are pretty low, 6, and then they jump to like 8. So this is an example where I think it's only 8 genes, but there's 100 of them at least that follow that same pattern. And when I cluster them, you can see these guys here, these guys there. These were done in May, these were done in June. And this is after normalization. So, getting back to this paper claiming Asians and Japanese people have 70% of genes different, this picture is showing us that. So we take each gene, and we have, this study it's a hat map, so it's probably like 90 whites and 90 Asians. You compute, for each gene, you can compute a p-value of testing the hypothesis that the null hypothesis are different. And if nothing is different, then this picture should look flat. This should be uniform, right? Because there's nothing to be found. There's all the p-values should be just uniformly distributed, and we see this big peak saying 70% or so of the genes are different between whites and Asians. Okay, well, look at what happens when we take, when you look at the dates, fortunately for everybody, the date of the processing is in the file with the raw data. So you can extract it and look at it. And in this study, the whites were done in one time in 1 year, and the Japanese were done in another. They were completely confounded by year. If I take just the whites and I separate them by date, I get a similar picture. So what is this difference? Is it biology or is it batch? I'm going to say I personally think it's batch because of this picture. So I don't believe that that's true. That different ethnic groups have just completely different expression patterns. And that's the end.